I wanted to impress on people in this talk that not everything is genetics. And when I say that, I mean not the genetic code. You can have genetically identical fish, but if you raise them in different conditions, feed them different things, you're going to get different results. And I've seen a lot of people start out with a line of fish and have pretty poor success with them early on and abandon them and assume that what the problems they're having are due to genetics and inbreeding and stuff like that. And I try to encourage them to look at other factors. It's like, what was the temperature that you incubated the eggs at? How old were the parents when you spawned them? Did you feed them, brine shrimp? A good example is, I think you've probably all seen goldfish with the operculum curls out. I think a lot of people believe, and there may be some genetic component to that, but I think 9 times out of 10, it's poor water quality or poor nutrition or high bacteria content. So you got to understand, you don't want to jump to the assumption that there's something wrong with the strain, that these fish are genetically flawed until you're sure that it's not you and what you're doing. Again, I think it's worth mentioning this. A good way to think about DNA, RNA, and proteins are DNA is your cookbook, it's your blueprint, and that just stays in the nucleus. RNA is sort of like if you open the cookbook and you write down a recipe on a piece of paper and hand that out to someone. Those are the instructions coming from your main log, and that's an RNA transcript. It's a short thing like that. And then translation will be the actual product, so whatever you're cooking. We talked about codons, which are the sequences of nucleotides in the DNA or in the RNA. There are 3 nucleotides long, and they each code for a different amino acid. So there are 22 amino acids, and depending on the sequence that you link them together, you create different proteins with different properties. Some of these codons do not code for a particular amino acid, but they mean stop. There's a stop codon and a start codon, and that is just what tells what's called the ribosome, which lands on the RNA and starts linking together the amino acids, where to start doing this and where to stop doing this. So it's very much like computer science, and how they have the zeros and the ones and things. Points of control. So I think you've got to remember once again, DNA does not code for phenotype at all. All it is a bunch of nucleotides and codes for proteins. These proteins, the way they get linked together is thermodynamics. It's like magnets and stresses and things like that. And you can manipulate that with things like temperature and diet, and that's why the temperature that you incubate eggs at is so important. We normally recommend, I would say, generally 68 to 70 is what I usually incubate eggs at. I've had some outdoor spawns where the temperature shot up to 95, and I've had some crazy, and I have pictures in here, crazy results, like partial dorsal fins coming out of those fish. What should have been double-tailed fish all had single tails, and it's just because under different thermodynamic conditions, the cell divisions occur differently, and everything that happens at the very beginning in the, when you have 20 to 100 cell stage, then those get magnified into bigger and bigger problems. Again, we talked about transfer RNAs and how they link up the amino acids to make the protein.
Will the temperature affect the sex of the fish at all, with fish?
Yes.
I know with chameleons, I used to have that, you can get more males and more females.
I think particularly with goldfish, and not all fish have the same sex determination system. for example, guppies actually have a true XY sex chromosome, and you see what's called sexual dimorphism in guppies to an extreme, where the male is obviously very different from the female. Goldfish, which are very difficult to distinguish the sexes of, you can change the sex ratio with the temperature. By increasing the temperature, you get more males. Now, it's easy to cause fish to develop ovaries versus testes by exogenous administration of hormones or affect it with temperature. They may have different chromosomes in there, but you get, like there was a thing that in the Potomac River, they were catching male bass that were producing eggs, and they were male by their genotype, and they were saying it may be from hormones in the water caused from humans, using birth control pills and things like that. So it's not as clear cut as you might think. The body can go ahead and start, once it gets on the path of developing testes or ovaries, that's the type of cells that it's going to produce, but the chromosomes in those cells may be something different. And occasionally we do find fish that are called its, that they might be beautiful fish, but every time you spawn them, you never get any viable fry, or they don't spawn.
Not spawning is pretty common. I had an experience one time where I was doing Chinese ranchu, and they were gigantic. They were about this big. They had about 30 of them, and they never spawned. And when we dissected the couple of them, they all had fatty livers.
Mm-hmm.
And so you can feed goldfish the wrong diet. In that case, I was experimenting with Ziegler's trout chow, and I was feeding the fish like 12 or 14% fat content in their fish food. That's their pellets. I just jammed them full every day of this trout pellet that was 12% fat, and they all got fatty livers, and none of them ever bred.
I also, through years of observation, and I don't think this is just a selection bias, my ranchu, I always get more females than males. And in fact, I usually send 6 out to people when they ask for a group of ranchu. Several times people have told me that they were all females. And the males were hard to come by. And I had the opposite situation with my Azuma nishiki, where I get many more males. And it could be a bit of a selection bias as you're culling. I like the big blocky, thick-looking ranchu, and maybe I'm inadvertently selecting for females that tend to be a little bit bigger. And then with the Azumas, I kind of like the long body and early color development, and maybe that's a male thing in those. But hedge your bets when you're doing your culling, save a little bit of a few different types. And particularly with something like the ranchu and line-bred strains that can be difficult and prone to breeding problems, if you have a little male that has tubercles and chases, even if he's not the best, keep him around, just because you don't know what the ones you're hoping for, if they're even going to be fertile. And I've often found that once that one teaser male gets going, a lot of times the whole group will start spawning, but without that fish in there, they didn't do anything. And my Azumas and even something like a Ryukin or a fantail, they seem to be a little more vigorous as far as spawning. So if I want to, I'll take one of those and put it in my ranchu pond, and it just seems to make a switch go, and they all start spawning, and then you've got to hand spawn the ones that you want to particularly cross. Okay, and this is a good thing to think about. How is this possible that a monarch butterfly has the same DNA, the same genome in all these different phases of its life, yet it's expressed so differently. It's like, how can you have the same organism look like that for a long part of its life, then look like that, then look like that? And the thing is, it's the genes are the same, it's just a matter of the timing of the expression and different environmental factors. Maybe it's feedback mechanisms where you get a buildup of a certain protein, and then it starts feeding back and shutting off expression of other proteins, and it's just the relative amount that trigger these huge phenotypic changes. So if that can happen in a planned way, you can imagine how much could go wrong with just, careless water changes and letting your temperatures swing wildly during the development of young fish. And let's see. Yeah, it has a lot to do with things like pituitary and environmental stimulation. Sunlight affects the pituitary, secretes different hormones that bring on some of the changes in that example. You also have a similar pattern in the metamorphosis of a tadpole to a frog. And this was an example. These piglets are identical clones, so they're 100% the same genes. They took an embryo and broke it up, so each cell has exactly the same genome. And the piglets were born, and you can see that although they're genetic clones, the phenotype isn't 100% identical. You do still have, like Gary was talking about, the migration of melanophores. That's the same thing happening here. You have similar patterns, but slight variations in where the piglet was in the uterus and where gravity was, related to the developing embryo, could have an effect. So you get a little bit of variation. Another interesting thing, I don't know if anyone's familiar with calico cats, and that only females are calico, or extremely rare for a male to be. And if you wanted to know why that is, it's because of a process called X inactivation. So here's an example. So you have a male genotype is XY. So you can have an orange male with the XY genotype. The orange color is carried on the X chromosome. Or you can have a black male. Again, the black is carried on the X chromosome. So those are different alleles. And then in a female, you would have XX. That's the genotype for female in mammals. But because you don't want double expression of the X chromosome, in males you only have one, so we talked about copy number. So the way that's countered is one or the other of the chromosomes, and this happens at a cellular level, not at the level of the organism, is inactivated. So you're only getting expression of one X chromosome per cell, not both. So depending on which chromosome gets inactivated, that cell is going to either produce melanin or the orange color. So you end up in females getting this spotty pattern of both orange and black being expressed depending on which of the two X chromosomes is active in that group of cells. Talking about fin growth, and that it happens, it's called saltatory growth, where the addition of a length of the fin happens in little bursts, and there you can see these little growth plates on the rays of the fins, and if you look at your fish's fins, you'll probably see those. And I think when we see twisted fins in fish, what it tells me is that there was a rate of change in the growth at some point. It could be nutritional, it could be water quality, it could be temperature, but you can imagine if you start growing and then you start slowing down and then you start growing fast again, you're going to have a little kink or a weak spot in that fin ray, which gets magnified as the fish gets longer. So during these periods of rapid fin growth, it is important to keep those temperatures as stable and consistent feeding and good water quality. Otherwise, you might get more of those deformities. These antifungals that people sometimes use on their eggs, their mechanism is to go inside the fungal cells and stop the DNA replication. And if it's doing it in the fungal cells, it could also be happening in the fish's embryonic cells. So some of these same things were used as anti-cancer agents, some of the dyes, under the same principle where they attacked rapidly dividing cells and disrupted DNA replication. So be careful about using any kind of dyes or antifungals. You're better off keeping the water clean, keeping them well aerated. If there's low fertility, you're going to get fungus. You do try it as a last ditch effort, but unless you absolutely have to, you got to consider that it could affect the development of the fish. Another thing is have a good spread. We do a lot of hand spawning. Use a large enough surface area. Try not to let the eggs get too clumped together. If each egg has a little zone with nothing touching it, there's less chance that the fungus is going to spread from egg to egg. So some scientists worked out the genetics of twin tail evolution. This was a paper that you shared, I think, Gary. Mm-hmm. And I don't recall exactly why I put those in there. I mentioned the curled operculum phenomenon and how that is likely an environmental problem. Temperature is the most important environmental factor. Small ponds and aquariums, the fish and the eggs are typically more subject to large swings in temperature, and I think trying to raise fry in tubs has really helped me keep things normalized. In my garage this winter, it got very cold, but the Intex pools that were on the slab, the temperature stayed pretty much the same because that concrete slab sort of normalized everything. The aquariums, say 20-gallon aquariums up on racks, their temperature fluctuated wildly because of the outdoor temperatures swinging at night and my space heater not keeping up, and I lost a lot more fish in those things, even though it was more the changes in temperature than the absolute temperature. Just keeping them constant was important. We talked about sex ratio, and higher temperatures can cause faster muscle growth relative to bone. You might see more spinal curvature and jaw deformities if the temperatures are too high. You do get faster growth, but sometimes a little bit slower growth at certain points in the development will give you a more uniform fish. If you push them real hard at the beginning, they can get real big bodies, but then as they get older, the fin size relative to the body size may not be ideal. And that may vary breed to breed. You may have to learn your breed and how you're most successful producing the best adults. Is it to grow them fast as they're little and slow them down? Or you were talking about with the telescopes, to keep them in shallow water and kind of go slow until the tail set is set, and then you can start.
Yeah, I'm going to cover that and when I go over the telescopes.
Okay.
I actually have a really good photo of something that was pushed too hard as a youngster, opposed to one that was given a little more time to grow out and get a proper tail set before I pushed it. So
another thing, when you're culling, I think it's natural for a lot of us to get excited about the big ones, the larger fry. It's like, this one's going to be great, but don't get too enamored with an oversized fry that's bigger than all the rest. There may be something else going on there. You can keep them around to see what happens, but don't cull all your smaller ones. They might end up being the better ones. Other things, environmental factors include light, photoperiod, dissolved oxygen, salinity, pressure, shear forces, pH, and exogenous steroids. Not that you'd have to worry about all those things, but they can happen. We talked about egg size. The embryo does start developing before the fish hatches, obviously, and then right after it hatches, during that phase where they're hanging on the glass, they need to have ample nutrients for cell division coming from the yolk in the egg. So that's why Oya or well-conditioned fish often have better looking fry. The first spawns from little toe size, you might find that the fry are weak, they just lay on the bottom, or they hatch out real early and they're just not viable. Don't give up on the fish, they're just still young. Let them go through a winter hibernation and load them up with gel food and try them the next year when the female's really ripe and the eggs are going to be of a better size. So phenotype induction, there can be something that happens at a particular critical time. For example, as the embryo is developing, the formation of the twin tail, at some point in there's a cell division that determines how things proceed from that point on. Whether or not that happens within a critical window might determine whether you get a fused tail, a single tail, or a double tail. So again, getting back to understanding your line, what the best environmental conditions are, and not varying from that. Calling that the scaffold that the rest of the fish develops on. And here are some of the azumas that I had that were born in a pond, and the temperature shot up to 95 degrees. It was during the summer. And water quality was good, but you can see they all have these dorsal spikes and single tails, which is not typical for the line at all when raised under ideal conditions. Observations from breeders, the water depth affects the body development, particularly in Ranchu and apparently in telescopes as well. Bloodworms fed early tends to encourage head growth in Ranchu and Aranda. Single-tailed fish develop folded tails when grown in too shallow water, so something about the water pressure helps stabilize the tail. And then obviously high temperatures produce more males.
Has it been established, by the way, the bloodworms, specifically the bloodworms, doing the Wen stuff, or I've been reading the Wen, I'm reading about protein and stuff.
I wouldn't say specifically to bloodworms, but
People swear by it, though.
they do. Bloodworms works.
I think there's other things that work.
Yeah, you got egg apparently works pretty well.
Eggs,
Steamed egg.
which are way cheaper than bloodworms.
What it gets down to is the availability of the right amino acids and lipids in the food, in the fish at the right time.
Okay.
The bioavailability of those versus maybe some of the flake foods and things like that might be much higher, and you're going to get better luck. Okay. And that was all for that one.